Himani ma'am,
Yes, sir.
you can start because I have started recording.
Okay, sir. Yes.
YouTube is also started, sir.
Okay.
Okay, sir. Good afternoon, respected attendees. I am Himani Ravale, and as a convener, I welcome you all on the behalf of Microbiologist Society India. As we all know, MBSI has started a PhD lecture series, and I welcome all of you for the very first lecture of the same. Let me welcome the judges for today's presentation. Dr. Nafisa Patel ma'am, Associate Professor and Head, Microbiology Department, Naran Lala College of Professional and Applied Sciences, affiliated to Veer Narmad South Gujarat University, Surat. And Dr. M.S. Ambawade sir, MSc, MPhil, PhD, Assistant Professor, Department of Microbiology, Haribai V. Desai College of Arts, Science and Commerce, Pune. Without further ado, let's start with the presentations. I welcome the first presenter for today's lecture, Dr. Abhijit Kare sir, Manager, Analytical Chemistry Laboratory, Serum Institute of India, Private Limited, Harapsar, Pune. His topic, expression of recombinant protein and its application in conjugate vaccine. Sir, you may start.
Yeah, good evening to all the members of Microbiology Society, and uh, my warm welcome is to Dr. Respected Deshmukh sir, and I'm thankful to all the members of society for giving me a chance to present my work in front of you. That's my PhD studies. So I need to share my screen.
Yes, sir, I have made you co host.
Fine, thank you. So as told by Imani ma'am, that's the topic of my today's presentation. This is expression of recombinant protein and its application in conjugate vaccine. To introduce myself, myself is Dr. Abhijit Karale. I am working at Serum Institute of India Private Limited from 2006 at a position of manager now in the analytical chemistry laboratory, which comes under the research and development wing of Serum Institute. Actually, this was my PhD topic when I completed my PhD thesis. That was transferrin binding protein (TBP) from Neisseria meningitis, and which was used as a novel carrier protein for conjugate vaccine. So this was my place of work where I completed my PhD work. That is, I was registered at Diva Patil Institute of Biotechnology and Bioinformatics at Tathawade, Pune. And my place of work was at my workplace that was Serum Institute of India. Dr. Nilu Nawani ma'am now she's the director of that institute, was my guide. And Dr. Raju Dere sir was the executive director. He was my co-guide during my studies. This was the objectives which I was proposed during my work and which was completed within the time frame. First objective was which was approved by RRC that time, and the first objective was to isolate, purify, and to characterize the recombinant protein, that was transferrin binding protein. The second objective was to explore the application of this protein, that is TBP, as a novel carrier protein in conjugate vaccine preparation. And after making this conjugate, there was the third study was to check whether this protein was really efficient in giving the protective functional antibodies when we conjugate to a polysaccharide. These were the three objectives which was approved and which was taken forward during the studies. So coming, starting from the basic, that it was my topic was conjugate vaccine preparation. So when we use a polysaccharide vaccines are not that much immunogenic because in infants they do not generate memory cells. Because the antigenic is a polysaccharide part, and we need to develop a conjugate which will produce a long-lasting immune response in infants. Polysaccharide vaccines, if we take in adults, it's okay; it will give us immune response, but to make a memory cells and strong interactions and to protect the infants, we need to conjugate this polysaccharide to some of the carrier protein. So it was a basic idea to make a conjugate vaccines, because nowadays many proteins are available, but that should be immunogenic and it should be safe and efficient in giving in the infants. So this was the basic idea to use the carrier protein in conjugate vaccine, because polysaccharide vaccines are not efficient in developing the memory cells and for long-lasting immune responses. So this is a basic chemistry reactions, means how the conjugate vaccines has been prepared or constructed. As the polysaccharide, we all know that is made up of different ring structures and with the pentose rings and all, and it contains a hydroxyl group, OH group. Ample amount of hydroxyl groups are present in polysaccharides. So we have utilized this concept of using this OH group by using a cyanolytic agent, that is CDAP. CDAP is 1-cyano-dimethyl aminopyridine. So what it does, it acts on the hydroxyl group of the polysaccharides, because it's long chain of polysaccharide monosaccharides. It contains a hydroxyl group, and when we react this polysaccharide with the CDAP, it generates a highly reactive cyanoester groups. And this cyanoreactive group is OC triple bond. You can see when it reacts with the protein molecule, because protein is made up of hydroxyl and amine groups. So the NH2 groups of protein have a very good interactions with this cyanoester, and they form isoavia derivative, which is a stable form of conjugate that we can say. So this is a basic reaction of conjugate vaccine, means how the conjugate can be prepared. So why we have chosen this topic, this protein as a carrier, because there was several, I earlier told that people earlier manufacturers has used CRM, tetanus toxoid, diphtheria toxoids as the carrier protein in many of the conjugate vaccines available in the market till now. But there are limitations of using this similar kind of proteins every time, because we can see that if we use a diphtheria, it requires, or tetanus, it requires detoxification, because they are toxins, and we need to convert the toxins into toxide. So there is a chemical reaction which use formaldehyde or formalin to detoxify this protein and convert these toxins to a toxide. So that is the main part, that detoxification is important. And during this detoxification, chances are there of reversal also. Sometimes in very few percent chances are there, it may reverse. The cases are very rare, but chances are there, but it may lose the some epitopic losses to the protein. That is the one lacuna was there for this detoxification process. Another is the bystander immune interferences and carrier-induced epitopic suppressions. So these are the two major responses what is happening when we use a similar kind of protein. They forms antibodies inside our body, and when it goes to a new conjugate vaccine, the antibodies against these for these proteins are already available, and some of the maternal transferation of these antibodies are also there. So there is already the crowd of that similar kind of protein is in our body. So when we use a new antigenic polysaccharide with a conjugated with this similar kind of protein, there is no interactions of the body doesn't find it that it is something new antigen is coming. So there is a suppression of the polysaccharides, because the crowd of antibodies against the protein is already there. So the polysaccharide which is antigenic, it gets suppressed, and we don't get an immune response for that antigenic polysaccharide for us. So these are the limitations for using of similar kind of carrier proteins. So to overcome this issue, there was a global need that its alternative carrier protein should come up for preparation of new conjugate vaccines, because the conjugate vaccines are ultimately a great need for the society. We need conjugate vaccines for various diseases available now, and we need to conjugate a polysaccharide to a protein for our infants, because it is, as I said, it doesn't generate B cell immune response in infants. So we need to conjugate anyhow that polysaccharide to a protein. So the transferrin binding protein has been proposed as a novel carrier protein in conjugate vaccine preparation. That was the main objective of our research study, which we have started. There's a obvious question will come back, why TBP you have chosen, because the TBPs are the outer membrane vesicles. So in my case, I have taken from Neisseria meningitis strain C. So I use this strain Neisseria meningitis as the protein from it is a OMB, and OMBs are already proven to have toll-like receptors and easily taken up by immune cells. And the beauty of this protein was it is generate both the humoral and cell-mediated immune responses. So that was the dual advantage of using this protein as a carrier protein, because it will give a dual advantage as an antigen also, as good antigen part, and as a good carrier protein also. So primary studies were already there to use DBP as a potential carrier vaccine, protein conjugate vaccines. But the further study was, and the characterization of this protein was a little bit on the lower part. So we have taken this topic for our study. And OMBs are also proven to a good adjuvant molecule. It gives a good immune responses when we immunize with when we conjugate with this protein. So it gives a good adjuvant activity also. So this was the basic 5 parts, which was my thesis was divided. That was the first part was upstream, that is a fermentation part. Second was the purification of this TBP, and third was the physiochemical characterization of this protein. And after that, thoroughly characterized protein was used for conjugation with the pneumococcal type 3. Initially was proposed for type 3, and 2 other were also taken further to make the conjugate of this polysaccharides. And after making that conjugate, we have done the immunological evaluations of this protein, whether this protein is really efficient and whether it generates the immune response when it goes to animal model. So at a glance, this was the 5 major milestones was there for this is my PhD thesis. So the first part was we started with the gene construct because it was a recombinant part, so we made a gene construct of it, and we have isolated this protein by using bioinformatics approach, and we have taken the sequence of that protein and we have transferred into plasmid, and it was further in E. coli cells BL21DE3, and upstream part was developed on that front. This protein was then further purified using membrane filtrations, column chromatography, and various other tools to make the protein free of other contaminants. When the protein was purified, it was characterized using various analytical tools, which will be coming in the further slides. And after this thoroughly characterized protein was used to make a conjugate against the polysaccharide with using CDAP chemistry. And this formulated conjugate was immunized into animal model in mice as well as in the rabbit. Evaluations of this immunogenicity study was done using IgG titers and MOPA, that is opsonoglycosis assay. So this is the protein, the sequence, which we can see that it's an N-terminal and C-terminal. It contains of 566 amino acids. It was taken from Uniprot, and the ID was A1KVG7. This is from Neisseria meningitis group C serotype. We have used this protein. This was the gene construct what we have made for our protein. It contains the red part, that was the cloning site where we inserted our protein. Initially, it was done with the his tag also, and after that we have removed the tag successfully. It was kanamycin resistance gene was there inside this plasmid. PET30 A plus plasmid was used to make this construct. This part was done with Gene Universal from the USA. We have given them a sequence and they have constructed this plasmid, and they have sent us that plasmid. And in here we transformed this plasmid into E. coli cells, and further studies was done in India. So the upstream part. Once we got that plasmid, we transferred into BL21DE3 cell line, it is the expression cell line, and it was grown in the LB broth with 30 microgram per ml of kanamycin. The single colony was again the single purified colony was taken and into a transformed to a 10 ml broth, and it was incubated for 37 degree at 220 rpm. Once we got the OD, we have transformed this fresh culture into a fresh LB medium, and it was grown further. Once we got the OD of 0.6 plus or minus 0.1, we have measured in spectrophotometer at 590, and then further it was when we got the OD of 0.5 or 0.7, we induced it with the IPT, that is the induction agent, to express our protein. So after 4 to 5 hours, we stopped that reaction and we harvested that cells, and it was centrifuged, and the pellet was taken into a buffer contain tris and ETTA. This was the microscopy which we have done during our fermentation part, so we don't find any contaminations at that time, and it was a pure gram-negative rod-shaped E. coli we got that time. So there was no contamination because already it has a kanamycin, so chances of contamination was less as such, but further contamination was not there. After that, we have done the total cell extract just to see whether our cells are really expressing our proteins or not. So we can see the 1, 2, 3 lanes are the non-induced cells, and 4, 5, 6 was induction, where induction was done. So they have expressed that protein at around between 70 to 55. So it was around 63, 64 kDa protein. What we have already, it was theoretical value, and which was matching with our expectations. So after the upstream part was done, we have taken this protein to purification. So because we have taken that pellet of that protein in a lysis buffer which contains EDTA and tris, and we have sonicated. Ultrasonication was done at 70 amplitude for 5 to 10 5 seconds with for 5 minutes with a 5-second on and off in very cold condition. We have used ice for that, so that the heat shock should not be given to a protein. And it was centrifuged, and the our protein was a cytoplasmic protein, so it went into a inclusion body because expression was overexpressed, so it was going into cytoplasmic form. So we have already also done a periplasmic construct and cytoplasmic construct. Both construct was done, but the yield in the periplasmic construct was very less. So we have chosen this cytoplasmic construct, and we have tried to express our protein with cytoplasmic only. So it was IB. So we have purified the IBs. So with the Triton wash was given because it reduces the endotoxin content from the protein, and the other impurities was also lowered down by using of these quaternary detergents. The wash of salt was given with 0.2 molar NaCl, and 3 more washes was given to that pellet with ultrapure water also to remove the unwanted moieties from the protein. So after this taking into a buffer, we have again same amplitude. We have given 70 amplitude ultrasonication to our protein to dissolve completely, and we don't gain any form of aggregation in our protein. And finally, it was taken in a dispersion buffer which contains a urea. Then the protein was further taken for purification. So we have done the fermentation. We have done the partial purification of protein till now. Then so we have, before loading into a column, we dialyze our protein with 15 because it contains the other components like urea, and also it may inhibit in our column chromatography. So we have dialyzed our protein with 10 kDa cutoff membrane and in a tris, and we contain tris and NaCl. In our study, we are using the Biorat microprep high Q resin. It is a strong anion resin. So this resin was used to purify our protein. So the column was equilibrated with the equilibration buffer, which contains tris and saline. Only tris equilibration was done with only tris. So we have continuously 10 to 15 column volume wash we have given to make the column properly equilibrated. And after that, we have loaded our protein with the minimal 5 mg per ml concentration, and it was allowed to react with the resin for some time. We have hold the protein in the column for some while. After that, we have taken the flow through of the protein, and the column was washed with the equilibration buffer with only tris pH 8.6, so that the unreacted, the bounded will be remain on the attached to the resin, but unbounded should come out, and the column should be free with the contaminants. And after that, we started elution with 200 millimolar and 500 millimolar NaCl, so that the protein which interacted with the resin is now started removing from that, and we collected the pure fraction of our protein. It was done primarily in the small gravity columns, and when it was optimized, it was transferred to ACTA purification system also. So we got the replicated results in both the cases. We got the same result in the small gravity column also, and the similar result was replicated in ACTA chromatography system also. So this is the ACTA picture. It was done on ACTA. So we have loaded our sample. The unbounded protein came into a flow through, and after that, thorough washing was given. Because the protein, the host cell proteins are also there, so the washing was given for a larger time to remove the unreacted moieties. So washing step was given more than 10 to 20 column volumes. After that, we started the elutions with our 200 millimolar and 500 millimolar NaCl. So we can see the peak of protein, and as different fractions of our protein was collected individually, so that the main region of a protein can be captured in a single tube. So this you can see that 3, 4, 5, 6 was a pure form of protein, but other was a little bit impure. There was a protein, but it was in low quantity, and little bit impurity was there. So if we speak commercially, it was a 5.2 mg was loaded. So this was a 4.65 what we got before chromatography, and we got a 1.82 mg of protein, purified protein after from the crude IBs, so around 30 to 35%. Yield wise, we can say we got the yield from the crude to a purified one. So now we have done the upstream part, we have done the purification. Now what the protein what we have purified should be thoroughly characterized. So we have done the different types of characterization studies. So this was a basic SDS page. We can see that the first lane number 1 was a crude form of protein that was expressed in a good quantity. Around 60 to 64 kDa protein was there. And after purification with this IB washing protocol and with anion exchange chromatography, we was able to get a single man in his dispatch. So that was, and I want to say that we have used only a single chromatography in our study. We have not used multiple chromatographies because earlier protein which was expressed by other researchers, they have used multiple chromatography steps like gel formation also, so anion exchange, cation exchange. But in our case, we have only used a single chromatography, that strong anion, and it was fortunately we were successful in getting a purified protein in a single chromatography system. So the yield wise and some labor wise, we can say that we can minimize the efforts in using this technique of chromatography or strong anion exchange. But the protein was there, but whether it is our protein or not, so we have done the Western blot of that using it is a transferrin binding protein. So it was having a high affinity with transferrin protein. So transferrin protein with HRP was available in the market. So we have used that as a to bind antibody was used to check whether our protein was which we get in SDS is really our transferrin binding protein or something else. But it was the interactions.
ये ऑफिस में है ये
So interactions was
UBS crude and
interactions was there with transferring HRP with the transferrin binding protein. We have done the SEC mols also for it. In this case, SEC mols is size exclusion chromatography with multi-angle laser light scattering. So the benefit of using this technique is to get the absolute molecular weight of our protein without any standard. It doesn't require any standard. It uses the different angle of that machine, and it gathers the information with the multiple detectors available, and it gives a absolute molecular weight without any standard. So it was around 64.36 what we got here. That was matching with the theoretical value what we have cloned in the E. coli. Basic HPLC like HPLC chromatography with size exclusion chromatography was used. In this case, in the first picture, you can see that crude IVs was there. It contains the impurity at 11.77 and also has 20.277. 18.8 was our protein of interest, but it was a crude IVs without any purification. Before doing the IV washing protocol and chromatography, it was that first picture. And the second picture, you can see that a single peak we got, and we was able to remove all the impurities from our protein, and the pure form of protein was observed after all the treatments. The standard active retention time was compared with the BSA. It has around 18 point something. So that RT was used as a standard for it. But it was just to cross check whether we are able to remove the impurities, because 11.7 is the aggregate part, which was we were able to remove with some of the ultrafiltration technique, and as well as the anion chromatography. Strong anion was able to remove all the other impurities like endotoxin and others. This is the enlarged picture of the earlier slide. You can see the crude IVs, and this is the purified form of protein. After that, we have after doing the HPLC, we have done the mass spectroscopy of our protein. The sample was with the help of Dr. Rahul Bambure sir. We have analyzed this sample at National Chemical Laboratory, and we have done the intact mass and peptide mapping of our protein. So it was around 97 to 98% homology of protein what we have cloned and what was expressed. So it was absolutely matching. Anything above 90% is acceptable. So we got around 97% coverage of clone versus express protein. So there was some missing sites, but it doesn't matter. But we got around 97% homology. Intact mass was also used. Intact mass we around it was matching with the expected values of 63.41 kDa, which was also given by mols and HPLC also. So intact mass by mass spectrometry give us the confirmation that the express protein is of our interest, and the other contaminants are not there. After that, mass with HPLCs, SEC mols, and SDS page and Western, we have done these interaction studies with Biacore technology. The sample was given to Cytiva, that's ZG chromatography system. They have a system, Biacore, which studies the interactions of protein with the ligands. So in this case, the dissociation and association was measured, and already said that this is a transferrin binding protein, and transferrin binding protein has high affinity with the transferrin, human transferrin or transferrin. So the sample was given, and along with the transferrin protein was also supplied to them, and we have asked them to do us experiment whether how the association are there with our protein with the transferrin. So this was the basic concept of interaction studies. They do emulation of halotransferrin protein into a gold chip, CM5 chip using amino coupling with EDC and NHS method. And in this case, the purified and partially purified samples was given to them, and they have the interaction studies were done with a 60-second association and 60-second dissociation. So that, and Biacore's T200 system was used with the evaluation software for that. So these are the some we have given this to them to a 0.2 molar elute, NaCl elute, 0.5 molar and partially purified. So 3 samples was given to them, and they have calculated the Ka, that is association, Kd, that is dissociation, and the capital Kd, that is the degree of association. Is how the protein is bounded to a transparent. So depending on that, the interaction studies was calculated, and the deviation percentage was absolutely matching with what was expected. So in this picture, you can see that the sample which is having first sample is a purified sample. We can see that was the plateau can be seen. It is association was very strong. There was no dissociation with them. Association was carrying to a larger extent, and dissociation was also slow in this case. So we can say when they start the association parameter, the association was constantly going on, and they give the dissociation parameters. It was dissociating very slowly. So it gives a good form of information that it has a strong interactions with the transferrin binding protein and transferrin. The other partially purified, the other
Sorry
form
to interrupt, but you have 5 minutes left.
oh, sorry. So this is
Yes,
the
sir.
other. So that's how the interaction studies were done. So in this way, we can say the 0.2 molar was a purified form. In this we got 7.92 constant, which the lower the value, strong the association. That is the main motto of this. We have done the gas chromatography, GCMS also. In this case, as this protein was, as I already said in first line, that it is a OMV, so it's outer membrane vesicle. So in this case, we want to prove this protein is it has some hexadeconic acids, so it has a good adjuvant activity. So we have confirmed with GCMS also. So these are the, at a glance, we have done the SD space, Western blotting, SEC MOS, peptide fingerprinting, LCMS, SPR, and GCMS. So using this, all the sophisticated tools, the protein was thoroughly characterized. So the further, after doing the characterization, we have moved to conjugation. For this, I had done some bioinformatics approach. We have published one paper on this. We transfer binding protein from Nigeria as a novel carrier protein conjugate vaccines. So we have completely done the bioinformatics approach, like modeling and doing all this, making the model, validating the model, finding the active sites of the protein into the model. And this was the bioinformatics studies. It was the model was validated with Ramachandran plot and PROSA analysis. And we have bioinformatically we have tested whether our polysaccharide, when we do the activation and whether it goes to bind to the protein. That docking studies was also simulation was also done. So these are the RMSD and RMSF values we have calculated. So we got a 2 stable conjugate into that. So this was the basic of this bioinformatics. So we have gone to a wet lab after getting some information from bioinformatics. We gone to a conjugate in a wet lab. So type 3 was used because it has a good, it has a huge demand in the market, and it is causing a lot of deaths every year. So along with type 3, we have 22F and 19F. These are the pneumococcal serotypes we have used for our studies. So CDAP is the basic chemistry. This is the chemical which what we use to make the polysaccharide activated. So CDAP was used for conjugating this protein. So this is the RI. This is the RI means refractive index, the detector. We got that is the size of polysaccharide. This is a protein. These are the two different parts. And when we make a CDAP reaction, it forms a conjugate. That is the third slide. So the conjugate was monitored for some time, for 3 hours, 4 hours. So we got a good conversion of polysaccharide to conjugate molecule. And when the conjugate was made, it was purified using different techniques, and it was analyzed to estimate the PS protein and free PS content using our wet lab tests. After that, we need to after the conjugate was prepared, it needs to be checked the protective immune response antibodies and whether it is immunogenic or not. So that time we have used this of the type 3, type 19A, and type 20F with CRM and TBP. We have taken the CRM as a control so that we should get confirmation whether our protein was really efficient against CRM or not. So by this was the machine IgG estimation method was used to the total IgG titers in terms of blood samples of from mice and rabbit. So and MOCO was also used whether it has a protective functional antibodies into that. So we can say that in our studies we got good results for all the 3 conjugates which were made with the TBP, and it was a good immune responses developed in mice as well as in rabbit. These are the graph pad graphs, and we can see that day 42 there was a huge increase in the IgG titers of 19,740 versus at 0 day we got only 50, but it got at 19,000 at 42 after, that is the post bleeding of final bleeding of 42 days. And his MOPA, that is optional phagocytic assay, we got that was a 4 at day 0 and 99, and any titer above 8 is a protective. So we got a huge number of titers in the mice model and rabbit model. for all the three conjugates what we have prepared. So these were the comparative studies against for the both CRM and TPP. So we got a good results for TPP against CRM. In both the cases, it was comparable for using, because TT is already proven, so it has a good heavy titer, but it was 72,000 is also a good titer. But the protective function antibodies in both the cases was equivalent, so that was the only considered. It is considered as a good immunogenic conjugate constructed. So this was the outcome of the studies, that we got a good response in animal, in both mice and rabbit. So some publications was done and conferences was also attended. These are some awards which I received during my PhD tenure from Microbiology Society of India, as well as from my initial I got a first prize at poster presentation for these studies. And I was thankful to all both the institutes for giving me this opportunity. And the study was successfully done in Serum Institute of India under the guidance of Dr. Rajiv Dere and Dr. Asha Malya, and my guide at D.Y. Patil University. And all my colleagues at Serum Institute was equally contributed in my studies. So thank you very much.
Thank you, sir. I request judges and audience to please ask the questions if there are any.
Hello.
Yeah.
Sir, that was very good in-depth work that you have been carried out. My only question is there any commercialization been done with your conjugate vaccine or not?
But right now it is under discussion, because we have done
Okay.
several batches with 2 liter and 5 liter fermenters, and
Okay.
we are able to replicate the results what we have done in shake flask, and they are
Mhm.
replicating in 2 liter and 5 liter flask. So we are under
Okay.
discussion with our higher management. Studies are also
Yeah.
going on. So I think so. I'm very much positive that it will be using commercial products in coming years.
This one, thank you. Thank you for your insight. And all the best for your future work as well.
Sorry, sir, you are not audible.
I didn't hear. Can you please repeat?
Yeah. Sir,
Can
could
you
you please write it in the chat box?
Yes, speaker is audible, ma'am.
No,
And
please.
is there any other question? Dr.
Yes.
Prakash
Is there
Rani is
any?
here. Dr. Nagrani is there. Any other question from audience? If there is a no question, you can conclude, Himani ma'am.
Yes, sir. Thank you, sir.
Thank you.
I request the second presenter, Dr. Steji Raffel, PhD from CSIR CFTRI Mysore, to please start a presentation. Her topic is identification of the biosynthetic gene involved in the synthesis of the peptide antibiotic of Bacillus licheniformis MCC 2514 and its application as a probiotic. Ma'am, you can start.
Hello. Am I audible?
Yes, ma'am.
So, good evening, everyone, and thanks to Microbiology Society of India for the opportunity given to me to present my PhD work here. So, shall I start with my work?
Yes, ma'am.
Actually, I'm not able to share my screen.
Yes.
Ma'am, have you made
No.
made you
First presenter has not deleted his presentation. First presenter. Please delete. First presenter has not deleted. That's why she is not.
I'll try to talk to him, sir.
Abhijit, please delete your presentation. You talk to him and tell him to detach.
Yes, sir. Ma'am, could you please try now?
Yeah.
Yes. Is it visible right now?
Yes, visible.
I'll start. Myself, Dr. Steji Rafael, and I'm here to present my doctoral research, identification of the biosynthetic gene involved in the synthesis of the peptide antibiotic of Bacillus urusheniformis MCC2514 and its application as a probiotic. The work was carried out at CSIR CFTRI Mysuru under the guidance of Dr. Prakasham Halami. So, this work explores a unique antimicrobial peptide produced by a probiotic bacterium and its potential use in functional foods. To set the context, I will begin with a brief overview of antimicrobial agents and probiotics before moving into our specific objectives and results. So, to the introduction, what are antimicrobial agents? We know that antimicrobial agents are substances that either kill microorganisms or prevent their growth. They include natural products such as antibiotics from fungi or bacteria, and also synthetic compounds developed as antibacterial drugs. Most clinically used antibiotics work by inhibiting bacterial, protein, or cell wall synthesis. Even though they have 5 different targets like DNA biosynthesis inhibition, RNA biosynthesis inhibition, cell wall synthesis inhibition, fatty acid biosynthesis inhibition, or protein synthesis inhibition, the vast majority inhibit either bacterial protein synthesis or bacterial cell wall synthesis. However, the resistance to these agents is a growing concern, and this has encouraged the search for new natural compounds with novel modes of action. So, one promising approach is to use beneficial microorganisms themselves. Certain microbes naturally produce antimicrobial compounds and can outcompete harmful bacteria. And probiotics are the heart of having such a principle. So they are living microorganisms that, when consumed in adequate amounts, provide a health benefit to the host. They help maintain a healthy balance of gut microbes, strengthen the intestinal barrier, and modulate the immune system. The common probiotic groups include lactic acid bacteria, bifidobacteria, and non-pathogenic E. coli, bacilli, and several yeast. The major characteristics of probiotics include acid bile tolerance, production of antimicrobial substances, safer use in food and clinical industries, persistence in human intestinal tract, and adherence to human intestinal cells. Among these, bacillus strains are particularly interesting because their spores are highly stable and can survive processing and storage conditions. Bacillus strains are widespread in nature and are found in soil, air, fermented foods, and the human gut. It has been demonstrated that spores of Bacillus probiotics can germinate, grow, and resporulate in the GI tract. And the commercially used strains include Bacillus subtilis, Bacillus closi, Bacillus coagulans, Bacillus urusheniformis, and several other Bacillus species are being used. So once we ingest a Bacillus probiotic, whether it is in a vegetative form or spore type, they will germinate or persist in the small intestine of the human body. And their major function is in the modulation of the intestinal microbiota by producing antimicrobial compounds, short chain fatty acids, or just a second, short chain fatty acids or organic acids or digestive enzymes. So when we talk about the bacteriocins, they are ribosomally synthesized compounds of a bacteria. They are bactericidal against strains of same or closely related species. And some of the bacteriocins produced by the bacillus include septilin, sublancin, mersacidin, coagulin, lichen, bacillocin, or bacitracin. And their properties include like their target can be membrane pore forming or cell wall biosynthesis inhibition, or they can also inhibit the peptidoglycan synthase. So we are particularly interested in a strain which is Bacillus aeruginoformis MCC 2514, which is originally isolated from sheep milk. And this native isolate has strong probiotic properties, also ATPase activity under low pH, cell hydrophobicity, autoaggregation, mucin binding ability, and cholesterol reducing ability. And it has the ability to produce a low molecular weight peptide antibiotic that is found to inhibit the RNA biosynthesis. And this probiotic bacteria was found to efficiently elevate the T regulatory cells, which are the anti-inflammatory cytokine producers. And it efficiently downregulated the elevated Th1 and Th2 cell-mediated immune response, which is classified as Crohn's disease and ulcerative colitis, respectively. Despite these observations, antimicrobial compounds that specifically inhibit RNA biosynthesis are not well studied, and no glycopeptides have been fully characterized from any probiotic cultures. So based on this, our objectives were formulated, which included purification and characterization of the IMC produced by Bacillus aeruginoformis MCC 2514, identification of the biosynthetic gene involved in the synthesis of the peptide antibiotic, and development of a probiotic product incorporating Bacillus aeruginoformis MCC 2514. Now we will move on to the most significant results of the study. So first, we confirmed the antibacterial activity and the mode of action of this particular antimicrobial compound produced by MCC 2514. So in figure 1, we can see that the antibacterial activity of the sulfur-free supernatant of MCC 2514 against the indicator pathogen Cucuria rhizophila, wherein the activity was not shown when it is treated with proteinase K. This shows that the compound is proteinaceous in nature, and the chromogenic plate assay using YVGS reporter showed a positive blue coloration for the 2514 and rifampicin. This confirmed that the target of this compound is the RNA biosynthesis. This was further confirmed through quantitative beta-gal induction, where a higher induction of beta-gal was shown by the CFS of MCC 2514 as well as the positive control rifampicin. Further, we did a media optimization study for the increased production of this peptide. Here, among the several media, an optimized tryptone yeast extract media was selected for the highest antibacterial activity at a fermentation duration of 20 hours. And later, we did a peptide purification using ammonium sulfate precipitation and butanol extraction. And the purification fold was improved from 3.490 in ammonium sulfate precipitation to 57.52 in the case of butanol extraction, with a percentage yield of 13.33. And with all the steps, we confirmed its antimicrobial activity as well as the mode of action using chromogenic plate assay was conducted. Later, we checked the molecular weight of this particular antimicrobial compound using trypsin STS phage and activity assay, and we found that the molecular weight of this peptide is around 2 kilodalton on comparison with the NICID against Cucuria rhizophila. So with the compound in hand, we moved to determine its potency and mechanism of action. Here we did kinetics of growth and peptide production. And we found that the antimicrobial peptide activity peaked at 2,400 AU per ml during the 20th hour of growth. And the MIC of the purified antibacterial peptide was found to be 6.3 and 4.5 respectively for the ammonium sulfate precipitated and butanol extracted protein. And we also checked the effect of this antimicrobial compound on the viability of the Cucuria rhizophila cells, and it showed that the IMP significantly decreased the viable cell numbers of Cucuria rhizophila in a time and dose dependent manner. And here we can see that the OD of the Cucuria rhizophila cells was specifically lowered to 0.097 in 24 hours at with the treatment of two times MIC of the butanol extract. Further, to identify the gene responsible for the production of this peptide antibiotic, we did whole genome sequencing of this bacterium using PacBio platform. And we found that the Bacillus uniformis MCC2514 consists of a genome with size of 4.23 MB and a percent GC of 46.2, and it had 4,386 genes with coding 4,276 coding sequences with 81 tRNAs and 24 RNAs, and these genomic attributes were compared with other available Bacillus uniformis strains, and this was comparable with the traits were almost similar with the other genomes as well. And this is the circular map of Bacillus uniformis with the gene coding region. And this whole genome sequence of the culture is deposited in NCBI under the accession number CP038186. Sorry. And the genome annotation was done using RAST, and the RAST annotation showed that higher number of genes coding for RNA metabolism, DNA metabolism, amino acids, and carbohydrates in our genome and compared with the other standard Bacillus uniformis genomes. It also showed that the genome contained 4 intact prophage regions and the probiotic marker genes for acid bile tolerance, adhesion, immune system modulation, etc. And it also contained biosynthetic genes for vitamins like biotin, riboflavin, thiamine, pyridoxine, etc., and also for essential amino acids. But no plasmid or virulence genes as well as CRISPR-Cas regions were found in the genome of MCC2514. Further, we analyzed the bacteriocin cluster present in the genome through Bagel4, and we found 4 clusters, namely sonoransin, lasopeptide, lichenicidin A1, and lichenicidin A2. And here is a bacteriocin operon analysis for all the 4 clusters with the structural gene being highlighted. Later, we did a bioinformatic analysis of these different bacteriocin clusters with respect to their molecular weight and mode of action. And we found that for lichenicidin A1, A2, and sonoransin, they have a similar mode of action wherein they targets the cell membrane. But in the case of lasopeptide, it is a special class of antimicrobial compound with a specific lasso structure, and they can have, and the reported mode of action for them are RNA biosynthesis inhibition, cell wall acting, or protein inhibitors. And we further detected the presence of these bacteriocin genes in our genome, Bacillus uniformis, and we found that all the 4 clusters genes were present in the 2514, and we compared it with the negative control, which is Bacillus uniformis 2512, which is also a probiotic bacterium, but with a cell wall acting mode of action. And to confirm the mode of action and to find that whether it is acting on the cell wall, we did SEM analysis. And the scanning electron microscopy revealed that the cells treated with our peptide remained largely intact, unlike those treated with the cell wall acting control peptide. And here we use the control as control Bacillus uniformis MCC2512, which is a cell wall acting IMP. And the treatment with that particular control showed an obvious breakdown of the cell structure, as we can see in the figure B. But there was no impact on the integrity of the cells when treated with the IMP of 2514 and with rifampicin. Similar result was obtained in the case of FTIR, wherein we can see that the MCC2512 displayed significant variations in the absorbance frequencies corresponding to lipid acyl chains, while the MCC2514 or rifampicin showed little to no variation from the control. So this suggests that the MCC2514 IMP does not specifically target the pathogen's cell wall components. Further, we checked the gene expression assay of Fukuria rhizophila in case of DNA gyrase and RNA polymerase, and this revealed a significant drop in RNA polymerase and DNA gyrase transcripts, and this shows that this further confirmed that the RNA inhibitory activity of the lasopeptide. So the mode of action study, genome sequencing, gene expression analysis, and molecular weight determination prove that the IMP is not cell wall acting but RNA biosynthesis inhibitor, and this suggested that the IMP is a lasopeptide. So we further analyzed this lasso cluster in detail, and we did a functional annotation of genes in lasopeptide cluster, and we found the biosynthetic precursor protein lasso and B1 and B2 protein, which helps in the processing of the precursor protein. And LASC gene is there to cyclize the peptide and the ABC transporters are there to transport this mature peptide. So the precursor protein will be acted by the B1 and B2 proteins and cleaves off the signal peptide, and this protein will be acted on by LASI gene, and this will cyclize the peptide into a mature protein, forming a S1D8 cyclization, and this mature peptide will get transported by the ABC transporters. We also checked the gene expression of less peptide genes at different fermentation hours. So we found that with increased time of incubation, the gene expression levels were also significantly improved for both analyzed genes related with the bacteriocin production. Further, we did a mutation study by applying a spontaneous mutation with ETBR on the Bacillus leishmaniasis MCC2514 cells. And we selected the mutants with no antibacterial activity, and we characterized these mutants. So among the 5 mutants, based on the RAPD banding pattern, only M2 and M4 mutants showed a similar banding pattern with the wild type. So these two were further taken for the sensitivity assay, and sensitivity was not observed for any of the mutants. So we analyzed the bacteriocin genes in this mutants along with the wild type, and we found that both the mutants M2 and M4 showed a significant reduction in the expression of the lesser peptide genes, and this further confirmed that the lesser peptide to be the gene responsible for the production of the peptide antibiotic. Further, we did a blast search and multiple sequence alignment of this LASO 2514, and we found that it is 77% identical to another protein, which is tricholamine, which is also a lysopeptide with potent antimicrobacterial activity. And this particular lysopeptide is produced by Streptomyces tricholaminecus. And based on the multiple sequence alignment, we found that only 4 amino acid, there is a difference of only 4 amino acids between the LASO 2514 and the tricholamine. So we checked the antibacterial activity of our peptide against Mycobacterium stigmatis, and this also showed a antibacterial activity against the Mycobacterium, just like the tricholamine. Further, we did the homology modeling for this peptide, and through ITASA, we predicted a model having a C-score of minus 1.91 for this LASO 2514, and this was further refined into more energy minimized structure using mod refiner and 3D refine, and we got a Q-mean set score of minus 3.21 for our structure, which was comparable with that of tricholamine and another lysopeptide microsin J25, and we got an ERAT score of 70, for the LASO 2514, and the structure also showed good stereochemical property with a probability confirmation of 63.6% residues in the core region of the Ramachandran plot, and there were no residues in the disallowed region of the Ramachandran plot. Further, we analyzed the potential active sites in the peptide through Cas-P3, and this showed through pockets in the LASO 2514. And the first pocket was having a higher area and volume, and this suggested that this can be used in the docking studies. So this is the first description of a lysopeptide from a probiotic bacillus strain, and this is the model visualization for the for LASO 2514, and this consisted of 176 atoms and 177 bonds. So this is the final model predicted for the LASO 2514. Later, as an application part, we developed a probiotic product incorporating this Bacillus uniformis MCC 2514. So we know that the Indian frozen dairy products are higher in demand by majority of the consumers, as it is quite cheap, palatable, and nutritious. In recent years, growing health consciousness led to the development of novel dairy products having therapeutic and nutritive value. The millets are globally recognized as the 6th most important grain, and it is an example of a staple food for people of lower socioeconomic status. Panicum sumatranus, which is known as little millet, is a good source of B vitamin, minerals like calcium, iron, zinc, and potassium. So based on this, we formulated a probiotic-based food product by incorporating millet as well. And we made probiotic millet kulfi, which can result in increased acceptability, value addition, and therapeutic value of the product. So the probiotic millet kulfi, we incorporated both little millet as well as Bacillus usualiformis MCC 2514. And this little millet is rich in potent antioxidant, tannins, and flavonoids, and it can help treat disease conditions like diabetes, cardiovascular diseases, cataract, cancer, respiratory conditions like asthma as well. So in the formulation of the product, we first washed and soaked the little millet, and we water extracted the milk from the little soaked millet, and we added palm jaggery, custard powder, and skim milk powder to this millet milk. And we boiled this milk until it thickens and allowed it to cool, and we added our probiotic culture into this. And we transferred it into molds and stored at minus 20 degrees Celsius for setting for almost 12 hours. And this resulted in the formulation of the product. This is probiotic millet kulfi. Further, we did microbiological analysis of this kulfi variants. Here we took the probiotic kulfi as well as the control kulfi, which doesn't contain the probiotic. And we also checked for the procured market kulfi for a period of 15 days. And the probiotic count was found to be 9.14, 1.2, 10 to the power of 7. In the case of probiotic kulfi with no E. coli or yeast-on mode contamination. But in the case of control kulfi and market kulfi, the contamination was found. In the case of probiotic viability, the probiotic viability of 8 log CFU per ml retained for 30 days in the probiotic millet kulfi. We also checked the proximate analysis wherein we check the moisture, protein, fat, ash, carbohydrate, and dietary fiber in the control kulfi as well as probiotic kulfi, which showed almost similar values. We also check the pH and viscosity of the different kulfi variants for a period of 30 days of storage. And in the pH, there was not much difference in the pH in the kulfi variants, while for the viscosity of the market kulfi was increased with the increased storage time, but there was not much significant difference in the case of control of the probiotic millet kulfi. We also checked the antioxidant activity in the kulfi variants, and for the probiotic millet kulfi, we found 79% of DPPH activity and 71% was found for the control. And in the case of total phenolic content, 79 and 89 mg gallic acid equivalent per 100 gram for control and probiotic kulfi respectively was found. We also did a color index analysis for the control and probiotic millet kulfi at different storage period, but there was no significant difference was observed between difference was observed until the 30 days of storage. The physical chemical analysis for the melting and cooling profile for the probiotic and control millet kulfi was analyzed, wherein we found that for the probiotic millet kulfi, the melting profile was 2.35, and for the control it was 1.13. And in the case of cooling profile, it was around 22 degrees Celsius for the probiotic and the control millet kulfi. We also analyzed the vitamins in the probiotic and control millet kulfi. And for the probiotic millet kulfi, we found the presence of the vitamins B1, B6, B3, B9, B2, and B7. And in the control millet kulfi, B1, B6, B3, and B7 vitamins were found. The sensory analysis also showed the probiotic millet kulfi obtained a comparable score in the sensory evaluation, stating its higher acceptance, which got an overall acceptability score of 8.29 with on a 9.8. Further, we did a neatly better preservation study using this probiotic culture. So here we added the probiotic cells as well as probiotic cell-free supernatant into the Italy batter. We mixed and stored it both at room temperature and at refrigeration. Here we check the changes in the properties like pH, carbon dioxide production, weigh separation, and odor of the Italy batter after the treatment with MCC 2514 in both room and cold temperature. Here, the bacterium has been shown to improve the shelf life of Italy better by preventing waste separation, CO2, and odor production while maintaining a pH of 3.96 to 4.29, especially at cold temperature. Also, we did a microbiological analysis of the Italy bacter, and this showed that there was no contamination in the bacterium probiotic incorporated Italy bacter until 6 days, both in room temperature and in cold temperature. So this study showed that the bacterium can be used both as an antimicrobial compound producer as well as a preservative. So moving on to the conclusion. So the genome mining of the culture revealed the presence of 4 putative bacteriosis in clusters, which are lichenicidin A1, A2, sonorensin, and lasopeptide. The mode of action studies proved that the AMP acts by inhibiting RNA biosynthesis, and the suggested lasopeptide is responsible for the synthesis of the antimicrobial peptide. The peptide was purified and the molecular weight was determined to be approximately 2 kilodalton, and the MIC was determined to be 6.3 microgram and 4.5 microgram respectively for the ammonium sulfate precipitate and the butanol extracted protein. And this is the first study to describe the biological activity of a lasopeptide from a probiotic bacteria. The homology modeling of the antimicrobial compound lasopeptide produced by Bacillus aciniformis MCC2514 concluded that this protein might be a better target to be developed as a drug inhibiting the RNA biosynthesis of the pathogens. The study was undertaken to explore the utilization of little millet incorporating probiotic culture, and a millet-based kulfi is developed. It was found to have a significant enhancement in the microbiological and proximate content of the kulfi. The results showed no significant differences in the sensory attributes of the developed kulfi. Biopreservation study showed that the bacterial culture incorporated Italy bacter didn't produce any contamination until the 6th day of the study, and we assure that the probiotic added batter can be stored in refrigerated condition for more than 8 days. This revealed the preservative efficiency of the bacterium and concludes that it can be used as a food preservative in different food systems. Thank you.
Thank you, ma'am. The questions are open now.
Hello. Hello.
Yes.
Yes, sir.
This is Dr. Mukund Ambode. Am I audible?
Yes, sir.
Yes, sir.
Wonderful presentation, ma'am.
Thank you. Thank you, sir.
A number of important things you are done in the number of concepts. Explain very well.
Thank you, sir.
Have you registered these have you registered for patent or utility patent
No, sir.
for your product?
No, we haven't done yet. We are planning to do.
Okay. This material or this overall content, can you use for development of other product, other probiotics?
Yes, yes.
Which are
Already.
regularly used, which are regular use in daily life.
Yes, sir. Already one
Consuming daily life.
is being developed using this particular probiotic carrot shreds, which is PDRE has been filed for that particular product actually.
Thank you. Wonderful.
Thank you. Thank you.
Good presentation. Very good. I'm very much thankful to Dr. A.M. Deshmukh, sir, President, Microbiology Society India, and convener of this program is Himani ma'am. Thank you.
Hello.
Yes, ma'am.
I was like just interested when you had that whole genome analysis of this particular strain. Could you also analyze if there were some genes for toxin productions or antibiotic resistant genes? Because this, yeah.
We didn't find any toxin or like virulence genes or any antibiotic resistant genes were found in this bacteria. We checked for that. It was not there.
Because at times this is what they may be opportunistic pathogen for the strain.
Okay.
And yeah, the molecular weight of your lasopeptide that you have determined. So were you able to correlate with whatever the bioinformatics studies that you have been carried out with this?
Parallel to this, another colleague of mine has done the peptide,
Okay.
The peptide studies have been done by another colleague of mine. So I was concentrating on the bioinformatic part.
Okay, fine. And regarding the yield, if you were able to get some good yield of this particular peptide.
With this particularly optimized media, the yield was just 13, but we did a media optimization using like this RSM method we have done, and we got a higher yield.
That was a nice study. Thanks. Thanks for that. Thank you, Arvind sir, as well as Himani ma'am, for the opportunity.
Yes. Thank you. On behalf of MBSI, I thank both presenters, and I extend my heartfelt gratitude to both the judges, Dr. Nafisa Patel ma'am, Associate Professor and Head Microbiology Department, Naran Lala College of Professional and Applied Sciences, affiliated to Veed Narmad South Gujarat University, Surat, and Dr. M.S. Ambavre sir, MSc, MPhil, Ph.D, Assistant Professor, Department of Microbiology, Haribhai V. Desai College of Arts, Science and Commerce, Pune. And sincere thanks to Dr. A.M. Deshmukh sir, All India President, India, MBSI, for organizing this event. Finally, a huge thanks to all the attendees. Thank you.